Hello everyone. In this class, we'll try to learn about how transport across cell membrane occurs. And importantly, we'll try to learn about what is passive transport in this class. Fluid and electrolytes on either side of cell membrane pass through the membrane by various means due to presence of different channels and carrier molecules. Selective membrane permeability generates the difference in composition between intracellular and extracellular fluids. Transport of specific substances like drugs, chemicals, and hormones also influence cell functions. Try to learn some of the uh, meaning of these words. Permeability is the ease at which the substance can pass through a membrane. Permeable means a membrane is said to be permeable when, by virtue of having pores, it allows substances to pass through it. Permeance means it is a property of membrane defined as the extent to which the membrane allows the passage of substances through it. Permeant means it is the property of a substance defined as the ability of a substance to pass through a membrane. Passage of substances through membrane can be broadly divided into two categories. The direct passage without the involvement of the carrier molecules, where cell membrane is a lipid bilayer, hence the lipid soluble substances such as gases, fatty acids, alcohol, ketone bodies, aldehydes, and many small uncharged molecules pass through the membranes easily. Water molecules also pass through this membrane easily, and this is called as direct passage. Passes through the channels or carrier proteins. Water soluble substances such as electrolytes, glucose, amino acids do not penetrate the membrane readily, or they do it indirectly. There are two major means through which these substances are transported across the cell membranes. One is through the water channels formed by the integral proteins and by combination with carrier molecules in the membrane. Okay, so this is called as indirect transport. Protein channels in the membrane. Proteins that constitute channels are selectively permeable. They are tubular structures that connect the exterior with the interior of cells. The permeability of the channels depend on the molecular size, shape, and charge on the molecules. Types of protein channels. Broadly, these are of two types: ion channels and water channels. Ion channels are integral proteins that span entire width of the membrane. Normally, they are formed by several polypeptide subunits. Some of the examples for ion channels are sodium channels, potassium channels, calcium and chloride channels. So, mechanism of working of these ion channels. Along the length of the integral protein, an aqueous pore is present around which the polypeptide subunits are arranged. The ions pass through the aqueous pore from one side of the membrane to the other without entering through the lipid bilayer. A polypeptide subunit forms a gate at one end of the channel that opens in response to specific stimulus. Opening of the gate uh, channel allows the ion to rapidly move, maybe 108 ions per second through channel. However, there is a selectivity filter that permits specific ions to pass through the channel. Therefore, these ions are often very selective. So this is an example of one ion channel. Okay, so which showing the integral membrane protein in a bilipid layer. So this is showing the gate which is going to open when the channel gets activated, and this is a selectivity filter which is going to open to specific ions. So these are the different ion channels that we'll discuss. So they can be ligand gated or mechanically uh, gated, and some of the channels are voltage gated. Depending on the voltage, they open up, and some of the ion channels are always open, and ion transport occurs depending on the concentration gradient produced across the cell membrane. Water channels are aquaporins. So there are 13 types of aquaporins have been identified. Gating of ion channels. Some of the ion channels remain always open, and therefore they are called as non-gated channels. However, many ion channels are open and closed by gates. So there are three general mechanisms of gating: voltage gating, ligand gating, and mechanical gating. Voltage gating: change in the membrane potential beyond certain threshold value opens or closes the gate of ion channels. For example, ion channels in excitable tissue such as nerve and muscle are mainly voltage gated channels like sodium, potassium, and calcium channels, also present in pacemaker tissue in the heart. For sodium channels, the gate is located outer end of the channel. The gate closes the outer opening of the channel when the cell is at RMP minus 70 millivolt in nerve, minus 90 in muscle, and minus 50 millivolt in pacemaker tissue. When inside becomes less negative, the gate opens and sodium influx occurs that leads to first formation of the local response and later genesis of action potential. So this is example for the sodium channel and this is a potassium channel. Note that the gate is on the outer side uh, of the sodium channel, okay, and in the potassium the gate is always in the inner side. Ligand gating: the interaction of the channel with ligand, hormone, or neurotransmitter causes opening or closure of the channel. As the ligand is a chemical agent, so this gating is also can be called as chemical gating. Mechanism: the chemical agent binds with a specific receptor protein on the membrane and brings out the conformational change in the protein that in turn uh, directly or indirectly opens the channel. Nicotinic cholinergic receptor channel in postsynaptic neuromuscular junction is a ligand gated ion channel. Acetylcholine released at the nerve ending binds with these receptors and open ion channels, that is sodium channel, that generate the postsynaptic potential. Mechanical gating: some of the channels are mechanosensitive and they respond to stretch. Examples are ion channels in hair cell in the cochlea, vestibular apparatus, and stretch sensitive channels in the ventricular muscles. Stretch imparted on the membrane opens these channels. The many types of carriers, so which can be uh, uniport or symport. Okay, so uniport is a carrier protein that transport only one substance. For example, sodium channel transport sodium and potassium channel transport only potassium. Symport carriers transport two or more substances from one side of the membrane to the other in same direction. The transport of sodium and glucose from lumen of the intestine or kidney tubule into uh, epithelial cells through the same carrier protein is an example for symport mechanism. The substances are also said to be co-transported. So there can be antiport also. Antiport carriers transport substances in opposite directions, in which one substance is transported to the inside of the cell and other substance is inside to the outside. Substance transported in opposite direction by one carrier are said to be also counter transported. Some of the example for counter transport are sodium potassium pump, so which is operated by sodium potassium ATPase. It actively transports 3 sodium out of the cell and 2 potassium into cell for each molecule of ATP hydrolyzed. So this is the gist of how the movement across the cell membrane occurs. It can be active transport or passive transport. In passive transport, it mainly occurs by the osmosis, diffusion, and facilitated transport. Whereas active transport mainly occurs because of the protein pumps and also the vesicular transport is called as active transport. Vesicular transport can be endocytosis or exocytosis. Okay. So here briefly given about the types of transports: passive transport, active transport, vesicular transport, and transport across the epithelium. Okay. So active transport can also be primary active transport or secondary active transport, and transport across epithelia can be transcellular or paracellular transport. So here are some of the difference between active and passive transport. Okay. So active transport, usually the substances are transported against the electrochemical gradient, where in passive transport it is along with the gradient. So utilization of energy occurs with active transport. uh, Energy is not required in passive transport. Saturation kinetic is a feature of active transport since are they require carrier proteins, but in passive transport they may not be present. Saturation kinetics may not be present. Passive transport. Common features of passive transport. There are two common features of passive transport processes. These are: transport is downhill. In passive transport, the substances pass through the membrane from both the sides. However, net movement occurs down the electrochemical gradient. Transport transport does not require metabolic energy. Passive transport does not require utilization of ATP. Examples are simple diffusion, facilitated diffusion, osmosis, filtration, bulk flow, and solvent drag. Simple diffusion. Diffusion is the process of passive transport. Molecules move from area of higher concentration to the area of lower concentration. In simple diffusion, molecules move due to their random movement. The examples of substances that pass through the cell membrane by simple diffusions are transport of carbon dioxide, oxygen, urea, ammonia, and ions. Factors that determine the rate of diffusion broadly can be into two categories: the properties of substances and properties of the membrane. So, properties of the substances that determine diffusion through the membrane are mainly the concentration and electrical gradient of that substance and permeability of the substance through the membrane. The concentration of the chemical gradient of the substance is primarily responsible for the direction of the transport of the substance. Normally, a cell membrane is polarized with the negative interior and positive exterior. Positively charged molecules that diffuses from outside to the negative interior is said to be moving along its electrical gradient. Its diffusion from inside to outside is said to move against the electrical gradient. Substances move along both electrical and chemical gradients. Electrochemical gradient is mainly used for ions. For gases, concentration gradient is expressed as a partial pressure, and for water, it's hydrostatic pressure or hydraulic pressure. Thermal energy contributes to random motion. In liquid or gases, all particles are in constant and random motion due to the thermal energy. Unidirectional flux. When two liquids are separated by semi-permeable membrane, the number of particles in the liquid striking the unit area from each side of the membrane at any given time is proportional to the concentration of substance in that side. These particles succeed in passing through the membrane along the gradient, so which is called as unidirectional flux. It's proportional to the concentration of substance in the liquid. Net diffusion is the difference between the opposing and unidirectional fluxes. Permeability of the substance. Factors that determine the permeability are the lipid solubility of the substance. Permeability is proportional to their lipid solubility of a substance. Molecular size. Permeability is inversely proportional to their molecular size. The and also depends on the charge on the molecule. Membrane is polarized with its negative interior. Cations easily enter from outside to inside the cell. Temperature also plays important role. Rate of diffusion is high at higher body temperature. Also depends on the distribution of ions in the membrane. The number of protein channels or substance in the membrane determines the permeability of substance. Fluid pressure gradient also contributes uh, diffusion of the membrane. Properties of the membrane. The two membrane properties govern the diffusion, that is the cross-sectional area of the membrane and the thickness of the membrane. So, so this can be given by the Fick's law. The rate of diffusion of a molecule through membrane is directly proportional to the surface area available for diffusion and inversely proportional to the thickness of the membrane. These factors are governed by the Fick's law of diffusion. According to Fick's law, for a substance whose inside and outside concentration are respectively Ci and Co millimoles per liter, so the net rate of diffusion is given as uh, dA divided by T into Ci minus Co, where D is diffusion constant. So that is, the net diffusion is directly proportional to the area, inversely proportional to the thickness of the membrane, and it depends on the difference in the concentration of that substance inside to outside. Facilitated diffusion. When diffusion is facilitated by a carrier protein in the membrane, the process is called as facilitated diffusion. Also called as carrier mediated diffusion, as a carrier protein facilitates it. Typical example of facilitated diffusion is transport of various sugars in red cells, adipose tissue, skeletal and cardiac muscles. The facilitated diffusion is also a downhill transport that does not require energy. Features of facilitated diffusion. It differs from simple diffusion by four special features. The faster rate of transport, it obeys saturation kinetics, so there can be competitive inhibition, and these reactions are, this transport is very specific. The carrier mediated or facilitated diffusion allows the transport of polar or hydrophilic molecules at a much faster rate than the rate expected from the partition coefficient. In facilitated diffusion, the number of carrier proteins available determine the rate of diffusion. When all the available binding sites on the carrier protein are occupied, the system operates at maximum capacity. So this is called as saturation point. Many substances share same carrier protein for transport. When more than one such substance is present on the same side of the membrane, they compete to bind with the carrier protein. For example, transport of D-glucose across the epithelial membrane slows down once D-galactose is present along with it, as they share the same glucose transporter. Carrier proteins are specific for different molecules. Factors affecting facilitated diffusion. The major factor is the number of carrier proteins available for the transport. Facilitated diffusion is also operated by other factors like hormones. For example, transport of glucose by GLUT4 into muscles and adipose tissue is insulin dependent. So these are the some of the difference between simple diffusion and facilitated diffusion, where in simple diffusion no carrier molecule is involved. Here the carrier molecule is involved. So simple diffusion does not obey saturation kinetics, and the diffusion is linear always, where the facilitated diffusion has a saturation kinetics. There will be no increase in the diffusion once saturation is reached. So there will be no competitive inhibition in absent, but the substances can compete for the same carrier protein in facilitated diffusion. Rate of diffusion is slow here and it is faster in facilitated diffusion. Simple diffusion is not specific, whereas carrier protein is very specific, or facilitated diffusion is specific for that particular substance. So this is showing the gist of the transport summary, I mean, where it has differentiated simple diffusion, facilitated diffusion and active diffusion. So simple diffusion does not require any carrier protein, but the substances move from higher concentration to lower concentration. In facilitated diffusion, they use carrier protein and substances are transported from higher concentration to the lower concentration, and the transport is faster here. But the active transport, additionally with the carrier molecule, they also utilize the ATP, and the transport is here very rapid, and the transportation can occur from lower gradient to higher gradient. Osmosis. Osmosis is the process of movement of solvent from solution with a lower concentration of solids to the solution with higher concentration of solids when both the solutions are separated by semi-permeable membrane. The net flux of water, that is the solvent, through a semi-permeable membrane from solution of lower solid concentration to that of high solid concentration is known as osmosis. Osmotic pressure. The osmotic pressure is the minimum pressure applied to the solution with higher solid concentration to prevent osmosis. The hydrostatic pressure necessary to prevent osmosis of the flow of water is known as the osmotic pressure of the solution. Oncotic pressure is osmotic pressure in the body fluid exerted by osmotically active substances dissolved in the fluid. The osmotic pressure in the plasma is known as oncotic pressure, which is 25 mmHg. Okay, so here, so uh, the osmotic pressure can be defined as the amount of pressure required to maintain the same volume in uh, A and B compartment when the the concentration is high in compartment A. Terms used in osmosis. Mole is the molecular weight of a substance in gram. The osmole and milliosmole, the concentration of osmotically active particles is usually expressed in osmoles. The millimosmole is 1000 of 1 osmole. Osmolarity refers to the number of osmoles in 1 liter of plasma, and the other transport uh, phenomenon. Filtration. Filtration is defined as a process by which fluid is forced through membrane mainly because of the difference in the hydrostatic and oncotic pressures on two sides. Bulk flow, when filtration results in movement of greater quantity of water, the process is called as bulk flow. Solvent drag is during the bulk flow of water, it carries with it dissolved particles, and this phenomenon is known as solvent drag. So the different distribution ions, so this occurs mainly because of the transport of the cell membrane. So this is in brief about the transport of cell membrane, particularly the passive transport. Thank you.